The human brain is made of at least 100 billion nerve cells. These include neurons, which are the nerve cells that are specialized for rapid, long-distance communication, as well as the glial cells. Glial cells are support cells that facilitate neuron function. The term glia simply means glue, and this stems from our original view that glial cells just kind of held the brain together, and it was the neurons that did all of the active lifting. They're the ones who fulfilled all of the functions of nervous tissue. We're appreciating that this isn't completely true, and glial cells contribute to the function of neurons in more ways than we originally anticipated. In this lecture, we're going to go over nerve cells. We'll start with neurons, because when you get down to it, they are the cells that carry out rapid, long-distance communication. They are the cells that allow my brain to control my mouth, my vocal cords, and plan out the appropriate actions for me to tell you these words. They're also the cells that allow you to hear the words I'm saying and translate that into some kind of meaning. We will go over the morphology of neurons and how they communicate with one another. The second part of this lecture is going to go over all the different types of glial cells and their basic functions. We'll start off with neurons. Neurons have complex and intricate morphologies that allow them to receive diverse input and communicate over long distances with very little delay. If we were to look at a few neurons, which we see here, here's 3 neurons in the cortex. You'll see 2 of them look kind of similar. These are what we would call pyramidal neurons because their cell body has kind of a pyramid shape. The other is what we would call a stellate neuron because it looks a little bit more like a star. You got to use your imagination here. But those are 2 broad classes of neurons. Pyramidal neurons tend to communicate over long distances, and stellate neurons tend to stay local. Regardless of whether they're pyramidal, stellate, or something else, they all have the same 3 principal components. The first would be the cell body. Whenever you learn about mammalian cells in your general biology class, that's what you're learning about here. So everything that a cell does, the cell body is going to do. This has the nucleus, it has all of the organelles that you've learned about. They're all contained right here in this kind of round central region. Sometimes this is called the soma, which is just going to mean body there. So cell body is the center of the cell. Radiating out from the cell body, we have these long extensions, which collectively we call neurites. And the neurites come in 2 different types. There's the axon, and then there's the dendrites. These are the specializations for communication that neurons have. So they extend these dendrites over long distances, and everywhere along that dendrite they can receive input. They can receive input from other neurons, so they can collect information and come to some conclusion. For example, what we're looking at, words that we're hearing, what they mean, things that we're thinking about doing later on. And they can communicate using their axon. This is another long extension that's going to project over some distance. It's going to be much greater than dendrites, almost as a rule. And the axon is going to be the transmissive end. So think of the dendrites as the ears and the axon as the mouth. The dendrites receive input, and the axon is going to project to somewhere else and provide some sort of message. Both of these are going to display alternating electrical and chemical signaling. So in the dendrites, we're going to translate chemical messages to an electrical message, and the axons are going to translate an electrical message to a chemical message. So let's go over the function of these different parts. It's really all about communication and, of course, keeping the cell alive. So we start in the cell body. Here we're just we've zoomed in on a neuron. Here's its cell body. You can see some dendrites coming off. Here's the axon. And this purple signal here, this is what we call Nissl substance. Nissl substance is just a collection of ribosomes and their associated structures that carry out protein synthesis. So Nissl substance is what makes proteins. And if anything happens in life, it's because a protein did it. Proteins are the functional units of cells. A cell without protein is not a cell. It's dead. You'll notice in the cell body, we have dense staining of Nissl substance. There's a little bit in the dendrites, and it's noticeably absent in the axon. Protein synthesis occurs largely in the cell body, and that makes sense. We have our nucleus here. This is where we translate, where we transcribe, I suppose, DNA to RNA, so that we have our messenger template that we can use to translate into protein. In the dendrites, there's some Nissl substance, so we have limited local protein synthesis in the dendrites and no protein synthesis in the axon. This is an important point that will come up again in the class. So all the proteins in the axon that are used to conduct that electrical signal, they all originate from the cell body. They have to be made here and transported down. If that transport process is disrupted, axonal function is disrupted. If not corrected, neurons with impaired axonal function die. So the cell body is very important because it creates protein. It's also the site where we integrate all those electrical signals generated in the dendrites, because you'll notice all dendrites originate from the cell body. So all the messages that the ears are receiving here funnel into the cell body. And this is what allows the neuron to carry out its alternating electrical and chemical signaling. That's how neurons work. Here's two neurons hooked together in a very basic circuit here. This connection is what we call the synapse. Within the neuron, we generate electrical activity. To communicate between the neurons, or a neuron and its target, we use chemical messages. Here we can see across time the electrical activity in these two neurons. So our presynaptic neuron, the one that's communicating, the one that's releasing neurotransmitter, this fires an action potential. That's that robust depolarization that we'll talk about in a moment. That conducts down the axon and causes the release of neurotransmitter that creates a slightly delayed and slightly weaker depolarization. The chemical messages that carry this out are called neurotransmitters because they transmit information from neurons to some target. These are received in the dendrites, and of course there are going to be multiple synapses on the dendrites. So neurons can receive hundreds to hundreds of thousands of synaptic inputs. And these all funnel into the cell body, sum together, and they determine whether or not the neuron is excited enough to fire an action potential, which travels down its axon, where it releases neurotransmitters to communicate with its targets. So there's our three basic divisions of a neuron. There's the cell body, where the neuron keeps itself alive by making protein, and where it integrates input from the dendrites. The second major division of the neuron would be the dendrites, the receptive end, where they receive input. This collects in the cell body and is conducted down the third part, the axon. The axon is going to conduct an electrical signal down its length and release chemical signals along the way to communicate with its partner. Neurons are going to have variable morphologies. We saw pyramidal and stellate neurons, but there are far more diverse types of neurons than just that. We characterize the morphology of neurons based on their dendrites because these are so variable. First of all, the number of dendrites can vary. We could have unipolar or bipolar neurons that really just have one primary dendrite. And what primary means is coming off the cell body. As the dendrite branches, it forms secondary, tertiary, quaternary, etc. But the primary dendrites come off the cell body. Some neurons only have one. Those would be our unipolar or bipolar neurons. In this case, we're coloring the dendrites blue and the axon kind of this brownish color here. As mammals, we don't really have unipolar neurons. We instead have pseudo-unipolar neurons. They start off bipolar, and then in development, the dendrite and axon fuse. Minor detail, but not minor cells. These form all of your somatosensory neurons. So when someone taps you on the shoulder, or you feel pain when you bump your knee, it's all because of pseudo-unipolar neurons. Other neurons have multiple primary dendrites, and these are what we call multipolar neurons. They could be the large multipolar neurons that we might see as a projection neuron in the motor cortex. They could be these pyramidal neurons as well. They could even have far more elaborate dendritic arbors like we see in Purkinje cells within the cerebellum. So there's great diversity in the structure of dendrites. And what this tells us is really how much diversity of input does this neuron take into account. A neuron that's simply transmitting pain, for example, doesn't need an elaborate dendritic arbor. If it senses pain in its receptive field, if I get a cut on my finger here, I don't need to talk to my neighbors. I sense pain, I transmit it. Other neurons are going to be involved with more complicated tasks, like making sense of language. In order for you to know what the word sucks mean, you have to take into account all the other words in the sentence. This class sucks, for example, has a very different meaning than my vacuum doesn't suck anymore. In one sentence, sucks is a bad thing, and in the other, sucking is actually what you want. So we have to make sense of multiple bits of information. The neurons that do that are going to have a multipolar morphology. So neurons in the cortex are going to, as a rule, have multiple primary dendrites so they can receive multiple sources of input and integrate information. Other neurons that simply transmit information, for example, from the retina back to the thalamus or from our finger to the spinal cord, those are going to have much simpler pseudo-unipolar or bipolar morphologies. Dendrites also vary in terms of dendritic spines. Dendritic spines are just these little spiny protrusions that come off the dendrites. That's how they got their name. They look like little spines. Not all dendrites have them. Some dendrites are going to be smooth and what we call aspiny. Others are going to have all these little protrusions. See these little white dots coming off of this dendritic shaft? These are dendritic spines. When you see these, you should be thinking that this is the postsynaptic site, and it's a very flexible postsynaptic site, meaning that its size can change. We can eliminate synapses, we can grow synapses. We think of dendritic spines as being the sites of learning and information storage. If we learn a fact, if it becomes easier for us to associate information, it's because we grew dendritic spines and they got larger. And when we forget things, it's because of the removal of dendritic spines, the elimination of synapses. You can't do this on the dendritic shaft as easily. You don't want to eliminate the dendritic shaft here because you'll lose everything downstream of it. You'll be eliminating hundreds of synapses as opposed to just one or a few. So neurons that have dendritic spines are going to have much more variability in their synaptic connectivity. We also see variability in terms of axons. Some axons stay local. These are on what we call local circuit neurons, and other axons project over long distances. These are on what we call projection neurons. Axons can branch highly in order to innervate multiple targets, or you can have relatively focal one-to-one targeting. This axon is an example of a highly branching axon. Here's the soma, or cell body, and the dendritic arbor, or the collection of all dendrites. Because it looks kind of like a little tree, we call it a dendritic arbor. Here's the axon, and once it reaches its terminal site, it branches extensively. We can tell from this that this neuron is going to be targeting many other neurons in this area. The basis of how neurons work comes down to the electrical charge at their membrane, or what we call the membrane potential. So remember, neurons are carrying out their function through alternating electrical and chemical signaling. The electrical portion of this is just changing the charge at the membrane. So when you learned about potentials in your basic physics classes, we're talking about the same thing. Potential is still a separation of charge, a charge difference. Membrane potential would be the charge difference at the membrane. Every cell has a membrane potential, but not every cell plays with it like neurons do. When neurons are resting, or essentially doing nothing, they have a negative membrane potential, like all cells. Neurons, we say, will rest at about -70. Of course, this varies from cell to cell. When a neuron is active, no longer are we negative. Instead, we have a positive membrane potential. And this is what we see during an action potential. When neurons hit a certain threshold, when they rise up above -70 to get somewhere in the ballpark of -50 or -55 millivolts, so after 15 to 20 millivolts of depolarization, once they hit threshold, then they start to spike. So the top and bottom panels are showing us the amount of current that we're injecting into the neuron, and then we're seeing the membrane potential as a result. We can measure and control electrical potentials using patch clamp recording. That's what we're seeing on top here. We take a glass pipette and we stick it down onto a neuron. Here's the pipette, here's the neuron. There's the cell body, notice the dendrites coming off, and here's a zoom-in cartoon version. There's a glass pipette sitting on the cell membrane. All the electrical activity that occurs in the cell gets detected with this pipette and a little electrode inside. We can also deliver electrical activity. Negative would be hyperpolarizing currents. Positive currents are going to depolarize the cell. When we say depolarize, we mean we become more positive than rest. So depolarization could just take us up to -60, or it could take us up to 0 and beyond. Hyperpolarization makes us more negative, so that's these first two here. What I want you to notice, this red line, the threshold potential. When our depolarizing currents bring the membrane potential above it, then we see action potentials. And by delivering stronger depolarizing currents, we don't see stronger action potentials. They don't get any more positive. Instead, they increase in frequency. 1 spike, 2 spikes, 3 spikes. Each of those spikes is triggering the release of neurotransmitter. So rather than having stronger depolarization, we just have more frequent action potentials. More frequent action potentials spits out additional neurotransmitter that causes more robust electrical activity in the dendrites. So the difference between a gentle tap and a strong tap doesn't come down to how much depolarization there is during the action potential. It comes down to the frequency. With a stronger tap, my somatosensory neurons are going to fire higher frequencies of action potentials. They're going to spit out more neurotransmitter, and they're going to stimulate their postsynaptic target more robustly. I'm going to perceive that in my cortex as greater pressure. But all it is a difference in the frequency of action potentials. So the intensity of the stimulus comes down to the frequency, not the degree of depolarization. When we hit threshold, we depolarize all the way. We go positive, that's more than enough to stimulate neurotransmitter release. And we'll talk about how this occurs in neurobiology next semester. But the Reader's Digest version is as such. Within about 3 to 4 milliseconds, we're going to go from -70 to +30. We'll hyperpolarize a little bit and then we'll come back to rest. The reason that occurs is because of an influx of sodium, or sodium rushes into the cell. Sodium has a positive charge. You bring positive charge into the cell, you make a more positive membrane potential. Slightly delayed, compared to the sodium current, we have potassium currents. Potassium leaves the neuron. When potassium leaves, potassium has a positive charge leaves, and this makes the neuron more negative. When these currents turn off, we go back to rest. And that's the action potential in a nutshell. The whole purpose of firing an action potential, of course, is to stimulate the release of neurotransmitter at the synapse. So this is how we get alternating electrical to chemical. The electrical event would be the action potential. The chemical event is neurotransmitter release. Neurotransmitters are only released when the neuron is sufficiently depolarized. And by having the action potential be all or none, such that once you hit threshold, you're going to depolarize to +30, you guarantee the release of neurotransmitter. When we release neurotransmitter at the synapse, The dendrites convert that into an electrical event. All the electrical events in the dendrites will flow into the cell body and sum up. If we hit threshold, we fire an action potential. If we remain above threshold because of very strong input, we fire multiple action potentials. If we were to zoom in on a synapse and turn it into a cartoon, this is what it would look like. We have really three components. There's a presynaptic site, postsynaptic site, and in most cases a glial element that we'll talk about in the second part of this lecture. The presynaptic site is exactly what it sounds like. It's that initial part of the synapse. It's before the gap. So we have a presynaptic site, a little bit of space, about a 20 nanometer gap called the synaptic cleft, and then a postsynaptic site. So think of this as your axon and your dendrites. The mouth, the ears. When we depolarize, that's going to cause the release of neurotransmitter, so it's going to open up calcium channels. Calcium comes in, and that calcium is what causes proteins to change their function and stimulate the release of neurotransmitter. So we release a chemical signal out here that's picked up by other proteins on the dendrite. Those proteins bind the neurotransmitter, and when they do, they create little holes in the membrane that allow ions to move. Maybe sodium rushes in and we depolarize. Maybe potassium rushes out and we hyperpolarize. The response to neurotransmitter depends on two things. What neurotransmitter did we release? There's a bunch of them. And what receptor to that neurotransmitter did we stimulate? Glutamate, for example, is almost always excitatory, except whenever we stimulate type 2 or 3 metabotropic glutamate receptors. So the devil's in the details. In general, you should be thinking of glutamate as excitatory and GABA as inhibitory. These are the most common neurotransmitters in the brain, at least the forebrain. Glycine takes the place of GABA in the brainstem and spinal cord. So you got glutamate to excite, and then glycine and GABA to inhibit. So presynaptic sites is where we release. The postsynaptic site is where we then generate that electrical current. If we generate enough depolarizing electrical currents in the dendrites, that neuron fires an action potential and communicates with its partners. And this is how neurons work. This is how we carry out movements. So let's go through a very simple nerve pathway that's highly relevant to you. We'll go through the corticospinal tract. Let's break this down. Cortico-spinal. Cortico means cortex. We're talking about the cerebral cortex. Spino would be spinal cord. So this is how we carry out conscious movement. So all this hand movement that I'm doing here, this is carried out through my corticospinal tract. When I think of doing something, I can plan the action in my cortex and then communicate from there down to the spinal cord, from the spinal cord to the muscles, and carry out a movement. That's, of course, nothing more than an alternating pattern of electrical and chemical signaling. So there's two neurons we need to think of for this pathway. The first would be the upper motor neuron. Upper meaning it's on the top, so it's above a lower motor neuron. It's a motor neuron because it makes us move, like a motor does, and it's a neuron. So the name makes a whole lot of sense. An upper motor neuron is a neuron up high that helps us move. These are going to be found in a few different regions. Primarily, we can think of the primary motor cortex here. So this is your brain. Here's the front, here's the back. We're looking top down, so we're looking at kind of this view of the brain. The sort of reddish color here, this is showing us the primary motor cortex. Primary somatosensory is behind it. These both contain a body map. That's what we're looking at here. So there's an orderly arrangement of neurons that kind of makes sense. The neurons that are next to each other in the brain are serving body areas that are next to each other. For example, your toes are next to your foot's next to your leg is next to your hip, and then that's next to your trunk, and then you have your arms, followed by the wrist and the hand and the fingers. So that makes sense. We slap our face on over here because we have to turn our body into a single strip. So there's some limitations. We put the tongue down here. But there's an orderly arrangement. Eyebrows are next to the eyes, next to the nose, next to the lips. So depending on which neurons we stimulate here, that's what's going to determine which part of the body are we going to move. So when those neurons in the cortex are stimulated, whether it's primary motor cortex, could be premotor, supplementary motor, or primary somatosensory cortex, these project downward. Some terminate in the brainstem, that's your cortical bulbar tract. So I'm using that a lot with speech. Controlling my larynx, controlling my mouth, that's all going to be carried out in the brainstem. Controlling my body, that's going to be down in the spinal cord. So the cortical spinal tract is going to continue on down to hit lower motor neurons in the spinal cord. And those are exactly what they sound like. They're still neurons, they still help us move, but they're lower. They're not up here, they're down here somewhere. So from upper motor neuron to lower motor neuron, that's it. That's your cortical spinal tract. 2 neurons. If the upper motor neuron is stimulated and fires action potentials, it's going to release neurotransmitter onto lower motor neurons. If those lower motor neurons are excited sufficiently, they fire action potentials and then release neurotransmitter onto muscles to allow movement to take place. And this is what allows us to plan an action and carry it out. It's just that simple. It's alternating electrical and chemical signaling. Now, just as there's an orderly arrangement of upper motor neurons, we also have an orderly arrangement of lower motor neurons. So when we travel down our cortico-spinal tract in the spinal cord, there's still an orderly arrangement. Legs next to the trunk next to the arms, and these will kind of peel off as we go from top to bottom of the spinal cord. So near the top, the arm axons will branch over and innervate the lower motor neurons here in the ventral horn. There's a body map here too. Those that are a little more lateral, so here's the midline, here's the side. They control more lateral structures like hands, arms, and the trunk is going to be controlled in the most medial portion of the ventral horn there. So there's a map in our brain, there's a map in the spinal cord, and then that allows us to then recruit our muscles in an orderly fashion. So that's it for neurons. In the second part, we're going to talk about glia. There's a few review questions. I suggest that you look at those and make sure you can answer them. Before we end here, let's make sure we get the main point here. So the first thing that we need to do is appreciate the morphology of neurons. We should know that they have 3 parts. The first major part would be the cell body, or the soma. This is what keeps the cell alive. This is where we make our protein. It's also where we integrate those electrical signals from the dendrites. So the dendrites out here, we should think of these as the ears. These are the receptive end of the neuron. We get synaptic input from the axons of other neurons here. So here's a little axon coming in. This cell, of course, has a happy little axon that'll communicate with another neuron somewhere else. Could be local, could be far away. When we release neurotransmitter from the axon, that's picked up in the dendrites, and that electrical activity is going to funnel into the cell body. And if we hit threshold, we will fire an action potential down the axon, stimulating the release of neurotransmitter on our postsynaptic partner. So there's the basis of of synaptic communication. We've got electrical, and we've got chemical. And we'll alternate back and forth to carry out this function. Now, in the case of our cortical spinal tract, this would be our upper motor neuron in the cortex. Here's our lower motor neuron down in the spinal cord. And then we have a muscle over here that we're trying to excite. If we cause this muscle here to contract, then we move. And the motor neurons have done their job. Now we turn our attention to the glia. I like to think of glia as the parent and the neuron as the child. Children run around screaming and yelling, communicating, if you'd like to think of it that way. And the parent is there to worry about everything else. So while the neurons are specialized for communication, they're not specialized for dealing with infection or paying the bill, picking up glucose from the blood, in other words, so that they can generate ATP. They don't store glycogen. They don't do a whole lot other than generate electrical signals within themselves and pass chemical signals to their target. And this is great, but without the glia, neurons would die. Glia are there to support all of the functions of neurons. So they are the ones who keep that electrical and chemical signaling going. The first class of glia to talk about would be the myelinating glia. That would be glial cells that make myelin. Myelin is a fatty substance that wraps around axons in order to improve the efficiency of action potential propagation. And we improve it really in three ways. First, action potentials are much more rapid when myelinated. When the axon's myelinated, we have rapid action potential propagation. So here's our neuron here. There's the cell body. We have the dendrites. They're just called neurites in this cartoon for whatever reason. And here's the axon. Along that axon, we have these purple myelin sheaths. Between each myelin sheath, we have these areas called nodes. These are little 2-micron regions, 2 microns long, where we have ion channels. So this is where we can generate electrical signals. The internodes, or the myelinated regions, have no ion channels. And that's because the myelin wrapping around the axon prevents the movement of ions. Even if we had ion channels in this neuron's membrane, there'd be nowhere for the ions to move. Think of this as sort of like the black rubbery insulation around electrical wires. This allows you to touch them without getting shocked, because the electrical activity can't move through the black sheath, just like it can't move through our fatty myelin sheath here. So the electrical charge we generate in the axon stays in the axon. This increases the rate of action potential propagation. We'll talk about why next semester. For now, just accept that that's the case. And by removing ion channels from the axon, we're also going to just move fewer ions. By moving fewer ions, we have fewer ions to pump back across the membrane, so we don't have to spend as much ATP. This is the second improvement. We spend about 1/75th the amount of ATP in a myelinated axon. So because we don't have to generate as much ATP, we'll find out later, we're not going to generate as many free radicals and damage the cell. So this makes action potentials faster and cheaper. The third major improvement that myelination offers is that we can use smaller axons to conduct rapid signals. Because we've sped up our conduction velocity, we don't have to create giant axons like invertebrates do. Squids, for example, don't have myelinating glia. They also don't have a skull. So they don't really have the same space limitations that we do as vertebrates. So when they want to speed up action potential propagation, what they do is simply make the road bigger. That's all they do. They increase the diameter of their axons. The squid giant axon is in fact so big, it's about 1 millimeter in diameter. It's so large that we can see it by the naked eye, and this is why we first studied the squid giant axon to characterize action potentials. Our largest axons are only going to be 20 microns in diameter. So they're about 1/50th the diameter, which makes them about 2,500-fold smaller in cross-sectional area. So if we were to use the same logic as squids to increase conduction velocity, our heads would have to be about 2,000 times larger, and our spinal cords would be the size of great redwood trees. We wouldn't be able to get around, in other words. Now, this increased conduction velocity only occurs in the myelinated regions, not in the nodes. So this causes what we call saltatory conduction, where the action potential kind of leaps from node to node. Because at the nodes we have slow, active propagation. In the internodes, where we have myelin and no ion channels, we have fast passive propagation. So the action potential, the depolarization that is, leaps along the axon. And that's why it's called saltatory conduction. Saltare just means to leap. Compare this to an unmyelinated axon, where we have to actively propagate the action potential along the entire length. This saltatory conduction is going to be about 10 times faster. Now, the two types of myelinating glia that we have would be the oligodendrocyte and the Schwann cells. Oligodendrocytes are found in the brain and spinal cord, so they're found in the central nervous system. And these have a different logic than Schwann cells, found in the peripheral nervous system, because they have different problems to solve. In the central nervous system, our brain is encased in a skull, so we have finite volume. We need to conserve space, so oligodendrocytes create a much tighter and more compact myelin compared to Schwann cells that can kind of fluff up a bit and make larger myelin sheaths. The reason for that is that they're out there in the periphery. We don't have the same protection that we do in the central nervous system. We're subject to bumps and bangs, and we want to make sure that our nerves don't get damaged. Neurons are going to have very limited regenerative properties, so we want to keep our neurons alive. In the periphery, we wrap them in this kind of fluffy myelin so that when we bang into something, there's a little bit of cushion for the cushion. And in the central nervous system, oligodendrocytes create a very compact myelin that's going to wrap around multiple different axon segments for each cell, and they're going to conserve space. That's their job. Both of these are going to do the same thing. They're going to increase action potential, conduction velocity, metabolic efficiency, and that is going to allow neurons to communicate more rapidly while spending fewer ATP. The next class of glial cells to talk about would be the microglia. Now these aren't actually nerve cells because they're not derived from neural progenitor cells. They're derived from essentially blood cells in the yolk sac. So these migrate into our central nervous system early on in development and live there for the rest of our lives. Microglia are essentially the immune system of the brain. Because we keep the blood separate from the brain to avoid killing off neurons, usually we don't have infiltration of white blood cells, only in extreme cases. Most of the time, the microglia are there to carry out immune function, that is, remove damaged cells, clean up debris, things like that. So the microglia are going to have really two different morphologies. So although they're not derived from brain cells, they do live with brain cells. And when they're resting, they look very much like a stellate neuron. They have their little central cell body, and they have these little projections coming off. They look a whole lot like dendrites. And in fact, microglia probably do listen to synaptic communication and participate. But we'll just think of them as macrophages, because that's what they look like once we have injury. So over here on the right, you can see a much more amoeboid shape. So the macrophages bring in their extensions and they kind of round up. That makes it easier to migrate to the site of injury and clean up the damage. So we have resting and reactive microglia. When microglia are reactive, they're going to have more of an amoeboid shape, so they can migrate and consume whatever foreign matter or damaged cells they find. They're also going to participate in recruiting immune cells in the case of infection. So they're going to play a role in inflammation as well. The next type of glial cell that we'll talk about is the astrocyte. Now these kind of do it all. If we had to call one glial cell the parent, it would be the astrocyte. These are a diverse class of glial cells that seem to have a far more complex role in nervous system function than we originally thought. And they have multiple types of extensions. So just like neurons, they have kind of a central cell body, and then they have these protrusions, which can be a few different types. They have what we call infoot processes, which are going to extend and essentially wrap around blood vessels. So here we can see a blood vessel running through a slice of brain. The white signal here is showing you this intermediate filament called glial fibrillary acidic protein. You see this in astrocytes. It shows you some of where the cell is. This connection protein here, CX30, this is just kind of showing you where astrocytes are communicating with one another or their targets. So that one's not as important. Here we can clearly see, though, the outline of a blood vessel traced by the astrocyte end-foot processes. Other extensions might be perinodal processes or cellular extensions, that would be processes that are near the nodes. So oligodendrocytes are going to form the myelin sheath, and at those nodes where we're moving ions, well, we still need to clean up the potassium that leaks out, and we need to provide ATP. That's what our perinodal processes are going to do. So some of the extensions are going to communicate with blood vessels, others are going to communicate with axons. This allows us to remove glucose from the blood and send it on over to neurons so they can generate ATP and they can continue to move ions. If neurons don't move ions, they don't communicate. If neurons don't communicate, we die. So the perinodal processes are going to play an important role in feeding neurons. Then there's the perisynaptic processes. Perisynaptic processes are exactly what they sound like. So these are cellular extensions that are going to be near the synapse. So here's our presynaptic site, here's our postsynaptic site, and here's a perisynaptic process from an astrocyte, colored in green. When neurons are communicating with one another, they're releasing neurotransmitter. If this neurotransmitter isn't cleaned up, it will continue to stimulate the postsynaptic neuron. And now a signal becomes noise. Any good signal needs to have rapid on, rapid off. And the removal of neurotransmitter is just about as important as the release of neurotransmitter. Neurons can clean up some of their neurotransmitter, but for commonly used neurotransmitters like glutamate, GABA, and glycine, we have astrocytic processes to clean those up. So here astrocytes are going to support synaptic communication by removing neurotransmitter. So they have, for example, glutamate transporters that will pick up that excitatory neurotransmitter and keep the neurons from killing themselves. This way they don't get too excited. Neurons can excite themselves to death. And glutamate transporters pick up that excitatory neurotransmitter and prevent that from happening. Astrocytes can also release their own transmitters called gliotransmitters to communicate to neurons as well. So they do more than just clean up after neurons, but that is undoubtedly an important job. If we were to remove those glutamate transporters, what we see are really two things. The first thing we see are severe seizures in these animals. So here's an example of a seizure occurring. We can measure this with EEG. So on top we have a normal mouse that has glutamate transporters. On the bottom is a glutamate transporter knockout. Notice these large amplitude deflections of the EEG. That's showing us seizure. And during that we see convulsions in the animal. This causes the animal not to grow as well and to die sooner. So in part A, we're just looking at the growth curve of wild type mice with the open circles, the filled circles, glutamate transporter knockouts. So when astrocytes don't clean up glutamate, the mouse doesn't function as well. It has seizures, doesn't grow properly, and as we can see in part B, starts to die soon after birth. So the survival curve looks very different for mice that lack glutamate transporters. While it's important to release neurotransmitter, it's also important to clean it up. The last point that I want to bring up here is injury, because that's what this class is all about. Injury to the nervous system. When that occurs, glia respond. So there's two players here. There's the microglia and the astrocytes, and these are going to do two important things. They're going to quarantine the area and they're going to promote cleanup. Quarantining the area involves forming a glial scar. So it's a thick, fibrous connective tissue that's formed around damaged areas of the nervous system. Here we can see the glial scar forming just around the area of damage. So immediately next to that, we form this glial scar, and that prevents the diffusion of potentially harmful substances out of the damaged area and into non-damaged nervous tissue. So this keeps the neurons over here alive and protected against whatever may be going on the other side of that scar. The second thing that our glia are going to do is actually recruit immune cells to the site of injury and act as immune cells themselves. So by releasing inflammatory cytokines, we can recruit white blood cells to an injured area. And what we can also do is act as macrophages. So the microglia will consume, if we have a bacterial infection, fungal infection, or if we just have dead neurons. Microglia will help consume the foreign or damaged cells. Now, let's see what we got out of this. Again, there are review questions to go over, but let's see if we can just summarize the main points. Kind of 4 things that we need to think about. The first would be myelin. So those myelin sheets that wrap around axons. So here's our axon here, and we'll have 2 different types. Let's make this top one myelinated, so we'll wrap some layers of myelin around it. Something kind of like this. Now we're looking in cross section. Keep that in mind. So the myelin actually wraps around the entire axon. And then we have our little nodes in between the myelinated regions. What you should keep in mind is that we have our ion channels all in the nodes. The internodes are devoid of ion channels. So all of the charge, all that positive charge that comes in the cell during an action potential, these will come in through ion channels at nodes and then propagate passively, but very rapidly through the internodes. Once they hit the next node, we get more positive charge coming in. Just make sure we're fully depolarized, and that can propagate passively on down to the next node where we get some more charge coming in to refresh the action potential. This is going to be about 10 times faster than what happens down here where we have to propagate along the entire length of the axon because it's not insulated. So that charge that comes in, well, hell, some of it leaks out. This is a much leakier axon. Some of it's going to move within the axon, but some is going to leak out. So this makes it much slower and inefficient because we're spending a whole lot more ATP because we're moving more ions. So your oligodendrocytes and your Schwann cells are going to be your myelinating glia. Then we have our microglia that look a whole lot like little tiny neurons until we have some injury. Then they become reactive and look a lot more like a little amoeba here. That allows them to move to the site of injury. Let's say we have a little bacterium that has invaded. The microglia can kind of wrap around that and eat it, removing the bacterium from our system. Same thing could be true if we have neuron damage and we have little bits of neuron to clean up. The microglia are going to do that. So this is an important part of cleaning up injury and infection. The last one to talk about would be the astrocytes, also called star cells. That's what this means. No one calls them star cells. Don't ever say that. Astrocytes are going to have a few different extensions.
One would be the in-foot process that goes over and kind of wraps around our blood vessels. So this allows us to take out glucose, stuff that's good, and put in waste products, stuff that's bad. There's also perinodal extensions, where the astrocyte can take this good stuff like glucose and funnel it on over and feed it to neurons. So when neurons fire action potentials, astrocytes can detect that with their perinodal process and then feed them a little glucose, because you know they have some ATP that they need to remake here. They're moving ions, they got to pump them back out. They need glucose to make ATP. And then we have perisynaptic processes. These are going to be found near synapses. Here's our presynaptic site. It's where we're going to release neurotransmitter. And we got our postsynaptic site, where we receive neurotransmitter. Our perisynaptic processes are going to clean up neurotransmitter. And as we saw earlier, this is very important for preventing the hyperexcitation of neurons and the death of the organism. Now, in the case of injury, let's say that we have a little injury and some cells are, let's say, dying off, something like that. We want to quarantine off this area of injury because there's bad stuff happening. Astrocytes are going to form a glial scar to prevent all the happy cells over here from being damaged by whatever toxic substances are generated at the site of injury. So by forming the glial scar, astrocytes can keep those happy neurons alive and separate from the toxic substances generated during injury. Of course, we'll also recruit our trusty macrophages here to come to the site of injury and help clean up. As we'll talk about in later lectures, the astrocyte and the microglia are both going to contribute to inflammation. By increasing blood flow at the site of injury and recruiting white blood cells, that's going to help us really deal with this injury.